Good morning, Year 13. So today we're going to finish off our work on synapses and the synapse end of the neuron by learning about something called summation. Now we're going to just launch into this today. Summation is the general idea that one synapse firing will not be enough, usually, to trigger depolarization in the next neuron. Now, when we think back to our graph of the action potential, which looks something like this, you're going to find that in order to get up to depolarization, you first have to pass through a threshold. Now, that threshold is about minus 55 millivolts memory potential. Once you get above minus 55 millivolts, the action potential can't be stopped. It's a kind of an all or nothing response. It's the kind of classic phrase. You get nothing if you get below minus 55, at minus 60, nothing happens. So if you take a little bit of sodium ions in and you get up to minus 60, sucks to be you, nothing happened. Once you get above minus 55, it's going to rocket all the way up. Because once you've gone past that point, it will trigger the opening of sodium voltage-gated channels all the way along the neuron. And that is what's going to happen throughout the neuron, and you can't stop it. All or nothing. But at a synapse, the opening of one synapse, like we talked about before, with all the calcium ions coming in, the vesicles fusing with the presynaptic membrane being released, and all of that, once we get those neurotransmitters into their receptors on the postsynaptic membrane, that will really cause the intake of small amounts of sodium ions. Now, that amount of sodium ions will not be enough to trigger an action potential generally. Generally, you require more sodium ion channels open than one synapse can do at one time. And therefore, what you require is something called summation. Now, what we're going to do is we're going to build up the levels of neurotransmitter in the synapse. And summation is just the building up of more and more and more and more neurotransmitter. And there's two ways we can do that, and I'm going to talk about both of them. I'll leave that on the board, but summation is just the general idea that you need more neurotransmitter generally than one synapse can provide at one time. You're going to need to build that neurotransmitter up. Once you've got enough neurotransmitter, then enough sodium ion channels will open, and then enough sodium ions will be taken in, and then you'll have your action potential. So, there's two kinds of summation, two ways of getting sufficient neurotransmitter. Spatial summation is the first and most simple one. Generally, in fact, I think always, a neuron is not just synapsed, if that's a word, with just one other neuron. Generally, multiple other neurons will attach onto the same neuron. Okay, so what can happen is that a number of the different presynaptic neurons can share the same synaptic cleft. So if I was redrawing my diagram, I wouldn't just have one synaptic knob, I would have 3 or 4, all attaching to the same synaptic cleft. And if they are all activated at the same time, they can release enough neurotransmitter to create an action potential. If only one of them is activated at a time, it won't release enough neurotransmitter. But if I get all 3 or 4, then I will get enough neurotransmitter. In effect, what I have is multiple neurons working together. One reason why we need summation of this kind is to stop every single little touch on your skin or every single little bit of light activating your receptors, and therefore activating your central nervous system. So every time I feel even the slightest touch on my skin, it will activate a receptor there. But it's only when I press hard enough on a large enough area that I activate enough receptors to send enough neurons being activated to activate the central nervous system and to feel that. Let's explain that better. Basically, you want more than one neuron to be activated before it's going to trigger a whole load of responses in your central nervous system. If every time one neuron was activated, we had a response in the central nervous system, there'd be too much information coming in at once. So what you need often is more than one receptor to be activated at a time, so that more than one neuron is activated at a time. They will all then feed into the same part of the central nervous system and have spatial summation. So it looks like this. In this case, both of these neurons here, both the ones with blue on them here, which we're trying to activate, will be below the threshold, because each one only has one synapse being activated. So even though there's very, very, very small amounts of sensation being felt by whatever receptor is activating this neuron, it's not enough to activate the central nervous system, the relay neuron here. However, let's say both of these neurons were activated at the same time, both of them have an action potential, both of them released neurotransmitters into the synaptic cleft, then suddenly there is enough neurotransmitter, it will bind on, it will cause enormous amounts of sodium ions to be entering, and then we have an action potential in the next neuron. So, no, yes. Spatial summation. Two neurons in the same space or more activating at the same time, causing enough neurotransmitter to be released to activate an action potential in the next neuron. Whereas here, not enough neurotransmitter in either neuron. Fine. So that's the first kind of two kinds of summation. Spatial summation. The next is called temporal summation. Temporal refers to time. So it's basically the same neuron activating multiple times. So in this case, there might only be one presynaptic neuron, just like the diagram we drew before. If it's activated only once, we've said that won't release enough neurotransmitter to activate the next neuron, to activate the postsynaptic membrane. But if it's activated multiple times, one after another, that will release neurotransmitter after neurotransmitter after neurotransmitter from the synaptic knob into the synaptic cleft, and that will lead to enough neurotransmitter being released to activate the postsynaptic membrane. But only if we exceed the threshold value for an action potential. Incidentally, I apologise for the number of full stops missing on this PowerPoint. I've stolen it from somebody else, in all honesty, and so, yes, I should have fixed that. Please make sure all your sentences end in full stops, for the love of English. So get those notes down, pause me if you have to, but the basic idea is one neuron activating multiple times has the same effect as multiple neurons activating at the same time. Looks like this. If there are a low frequency of action potentials, you know, one every minute or so, that's going to release a little bit of neurotransmitter, but it's not going to release huge amounts. If we have extremely high frequency action potentials, that's going to release loads of neurotransmitter, pretty much all the neurotransmitter the synaptic knob has, and that will activate the action potential. And basically, this is how neurons control how strong a signal is. So the difference between just touching your skin and pressing your skin hard, so it hurts, is that if you just press it slightly, it's still activating the same action potential, it's still going up to the same levels, plus 40 and all that. But it doesn't release many new action potentials. If you press hard, you're going to release lots of action potentials, one after another. That's how your body knows whether you're being caressed on the face, punched on the face. Caressed on the face, just occasional action potentials. Not really enough to activate many other neurons, just enough to tell the brain what's happening. Punched in the face, loads of action potentials one after another, because the receptor is saying, I'm not just being touched here, I'm being touched hard. And so your body then activates other neurons because of temporal summation, including your pain receptors, fight or flight responses, adrenaline released, and all of that stuff. Because if you've got lots of action potential being released at the same time, you'll release lots of neurotransmitter, and it will activate loads of neurons. Fine, so that's temporal summation. That is the second of two types of summation. Never fear, there is a little bit more detail here to add, because we're always assuming here that these are positive responses. I'm going to touch on some negative responses and I'll show you what I mean. Before we do that, we're going to do some questions. 4 questions. Do I need you to write them down? Probably not, these questions. Have a look at them. Have a look at them, make sure you've got an answer in your head. If you think it'll be helpful to you, write them down. But pause me, make sure you've got an answer in your head, and then we'll go through. So, summation. Summation is, and it's a relatively difficult word to define, you will never be asked to define it in a test. Summation is the idea that you require multiple doses of neurotransmitter, usually, to activate an action potential in the postsynaptic membrane. So, one dose of neurotransmitter from one synapse will not be enough. You need multiple doses. And that can happen through multiple neurons being activated, or the same neuron being activated multiple times. Either way, releasing multiple waves of neurotransmitter to activate the postsynaptic membrane. The difference between temporal summation and spatial summation, the main one, probably the best way of putting it, is the number of neurons. Spatial summation requires multiple neurons, temporal summation requires only one neuron. The other difference is spatial summation only requires each neuron to be activated once. Temporal summation requires each neuron to be activated multiple times. Explain how temporal summation allows the postsynaptic membrane to reach the threshold value. Well, because each time an action potential happens, and bear in mind with temporal summation we're having lots, each time it happens we'll release small amounts of neurotransmitter, and each time we release small amounts of neurotransmitter, a few sodium voltage gated ion channels open. So if it happens multiple times with lots of neurotransmitter, enough sodium voltage gated ion channels will open to reach a threshold. And suggests an advantage of responding to high level stimuli but not low level ones. It removes the ability to overreact to low level stimuli. If every time someone touched your face, your body went through all of the reflexes that happen when you get punched, flinching away, heart rate increasing, hairs raising, it would be very annoying. You respond only to high level stimuli to avoid overreaction. It's probably the best way of putting it that I can think of. So, there's one more thing to touch on, and that is inhibition. Now, I know some people have already read ahead, have read about this. Some people, I think, are actually using their textbooks rather than watching these videos. I can't stop you. Not that you'll be watching this video, so you wouldn't know. But I know some people are ahead of this. I want to touch on it anyway to make sure everyone's got it. So, as well as sodium ion channels on the postsynaptic membrane, there are also chloride ion channels. Now, I hope everyone knows that chloride ions are negative. The clue's in the symbol Cl-. So what's going to happen if you take in chloride ions to the membrane potential? If you open your chloride ion gates and chloride ions rush in, you will hyperpolarize the membrane, i.e. it will become more negative inside. Now that will either completely prevent you from making an action potential, or at the very least it will make it much, much more difficult. So, if these chloride ion channels are activated, it actively prevents a neuron, an action potential from being propagated in the postsynaptic membrane. Now, why might you want that to happen? Well, think about it this way. When you put your clothes on for the first time in the morning, you can feel what they feel like. You know, you pull on your trousers, you can feel the fabric on your legs. You can feel if it's jeans, you can feel if it's leggings. Throughout the day, though, you don't constantly have that sensation. In fact, it would be very annoying if you did. If you were constantly feeling your trousers like you did the first time you put them on in the morning, that'd be incredibly annoying. Right? Basically, if you have the same stimulus happening over and over and over again, it's probably best for your body to ignore that. And the way it does that is that if the same stimulus is happening over and over and over again from a receptor, bear in mind your receptors are still being activated by your trousers even if you can't feel them, because they are being touched by them. But if they're activated over and over and over again, what happens is actually other neurons are activated by that number of action potentials, which release an inhibitory neurotransmitter called GABA. GABA, G-A-B-A, or a different inhibitory neurotransmitter. There are a few. We're not going to learn about any of them, to be honest, not even GABA. But broadly, they all work in a similar way. They're going to release chloride ions into the postsynaptic membrane. So it works in the same way. So my trouser avoidance neurons, if you like, the ones that stop me feeling it all day, they're activated by these receptors feeling my trousers all day. They send an action potential all the way along these inhibitory neurons, if you like. Those inhibitory neurons then release GABA rather than acetylcholine or noradrenaline or whatever it might be. They release GABA. That GABA, exactly the same way, calcium ions entering, GABA released into the synaptic cleft. It transmits itself by diffusion across the synaptic cleft, binds to a receptor, just like acetylcholine, but in this case, the GABA receptors are attached to the chloride ion channels. So when they are activated, chloride ions enter the postsynaptic membrane and not sodium ions. What that happens is then we then block whatever signals are happening. We make it more difficult. Now, if someone presses against my trousers and they press against me, I'm still capable of feeling that. Because what will happen then is my receptors will send more action potentials, more temporal summation, more acetylcholine, and that will overwhelm the GABA. That will cause so much sodium ions to enter that it really doesn't matter about the chloride ions, we're still going to have an action potential. It just means that stupidly low level or stupidly constant stimuli are ignored. Because the neurons are desensitized, if you like. They're turned off, if you like, by these chloride ions. And this is called inhibition, and it's the final idea we need to have about synapses. Once we've got the idea of how synapses work, which, if you didn't have that already, you're not going to understand this lesson, unfortunately. And once we've got the idea of kind of the idea of how medicine might work at the synapse, that's the most woolly bit. This is the final bit with summation. So, this kind of an idea. So a neurotransmitter opens the calcium channels. This GABA, let's say. No, sorry, that's ridiculous. Calcium opens the yeah, I'll apologise. This slide needs changing slightly. Broadly, with this synaptic knob up here, calcium ions enter through their voltage-gated channels, just like before. They cause these vesicles here with the green neurotransmitter in them to fuse with the presynaptic membrane, as they are. These green neurotransmitter dots are GABA. They are not acetylcholine. The GABA, which are now for some reason red dots, will then attach themselves onto these receptors, which then cause chloride ions to enter. Now, that will cause hyperpolarization. Okay, fine. To be honest, I think that diagram may have made it more confusing than it was before. Broadly, there's a new neurotransmitter. It's called GABA. It opens the chloride ions channels, not sodium ion channels, and it stops action potentials from working properly. Fine. Right. Now, as I say, it's not like one synapse will have an inhibition and one synapse will have a positive excitatory stimulus. They work antagonistically at the same synapse. So almost always the GABA neurons are being fired. It just depends then how much they're being fired, and it also depends how much temporal or spatial summation there is of the acetylcholine receptors. So it's a balance between the two. If you have a lot more acetylcholine than GABA, then you will activate the neuron. If you have a lot more GABA than acetylcholine, you will not have an action potential. Fine. So it's just another part of summation, really. Fine. Well, hopefully we've all understood that. Apologies for that slightly confusing diagram there. What we're going to do now is going to do a series of questions. Now I have to say there seems to be a tendency, when I'm looking at your notes, from some people to skip the questions at the end. Perhaps they've already paused and closed the video. Please do not do that. It is extremely important that you understand these topics, and I pick these questions because they're going to help you understand it better, I hope. Now, obviously it's entirely up to you, I can't make you. But let's go through these questions. So, these first two are just very straightforward action potential questions. Let me get rid of this little bar down here. So 312 is the answer. It's not much to do with synapses. It's the first part of a question which later on has quite a lot to do with synapses. So could we please pause me and have a go at these questions? Just the answers, there's no need to write out the questions or anything like that. So, let's have a look at the answers for this one. So the mark scheme for this one is slightly odd. Broadly, we need to have the general idea that there are proteins inside that are negatively charged. That's quite a nice, simple idea that's always true. The membrane is more permeable to potassium ions than to sodium ions. For that, you could talk about potassium leak channels, and I would, and you would get the mark for that. So that's basically the same thing. It's saying more channels for potassium than for sodium. Potassium ions diffuse out faster than sodium ions diffuse in. To be honest, there's not a huge amount of sodium ions diffusing in. There are very, very, very low levels, so that's definitely true. You need to mention the sodium-potassium pump. That's something everybody should be mentioning. The sodium ions are pumped out faster than potassium ions are pumped in. So fine. The 3 marks I expect you to get are for talking about the proteins. Never forget them. They're very important. For talking about the potassium ions diffusing out, or for talking about the leak channels, and then for talking about the sodium-potassium pump. People do forget about the sodium-potassium pump. It's the most important bit of all. Without that, this just doesn't happen. So that's fine. The question was, what makes depolarization happening? The sodium ion gates opening and sodium ions rushing in. Fine. And then part 2 here, why another action potential cannot be generated during repolarization, because the membrane is impermeable to sodium ions, or the sodium ion channel is closed. I'd expect you to use the word inactivated, but closed would get you the mark here, and therefore sodium ions cannot enter the axon, and therefore membrane becomes more negative than the resting potential, and that's fine. So, straightforward there. The first 2 marks are the ones that I'd expect you to get there. Fine. Let's keep going with a bit more synapse work. 2 marks and then 1 mark. Please pause me again. So, explain why a neurotransmitter can only bind with one type of receptor protein in the postsynaptic membrane. Well, this is just talking about your older structure and shape. Unique shape of receptor protein or binding sites. Active site they're going to reject, i.e. they're not going to give you that mark at all. Even if you've mentioned active binding site, let's say, you will lose that mark because active site is only for enzymes. Due to the tertiary structure of the protein molecule is another mark. And then the concept of a complementary shape or a reference to neurotransmitter fitting will get you a 3rd mark. So the receptor protein has a unique shape, which is complementary to the neurotransmitter, will do just fine. And you should really be talking about tertiary structure when talking about proteins. We should know that all receptors on the surface of the postsynaptic membrane are protein receptors, so we should know their tertiary structure. And then, what's the role of calcium ions? It causes vesicles to move to the presynaptic membrane and fuse with the membrane. Make sure you're talking about presynaptic, you need it for the mark here. Right, and then finally, some work on summation here. Another 6 marks. Now I'm aware there's been lots of questions here. These are the most important ones of all. Make sure you can do them, please. And pause me again. So, we're looking at a relatively complicated question here. There's a lot of application here. So in terms of summation, explain why an action potential is generated in neuron D in time period 2. So time period 2, we're looking at several different things here. So in time period 2, you've got two different neurons being activated, and each one being activated twice. So you've got both temporal and spatial summation. And the marks you need here are the impulses and action potentials from neuron B, A and B together, or spatial summation, that's a mark. Cause sufficient depolarization, or open sufficient sodium ion channels. So, or 4 thresholds to be reached. So whenever we're talking about the summation, we want to be talking about the amount of sodium ion channels that are open. Because that's the point. That's why we need a lot of neurotransmitter, is to open a lot of sodium ion channels. And the threshold is also a key point. If you're not using the word threshold at this point, that's what the summation is getting us. It's getting us over the minus 55 millivolt threshold. Right. Part 2. Explain why no action potential is generated in neuron D during time period 1. Well, you can see here there is apparent spatial summation, i.e. A and B are both activated, but they're activated at different times. So by the time B is activated, the A one will already have faded away, the neurotransmitter will have been broken down. So, impulses from A and B are independent or no summation would be the mark I would expect you to write. Threshold not reached. Again, that idea of threshold is key here. And again, the idea of the sodium ions is also key. Whenever you're talking about summation, you're talking about levels of sodium ions entering the cell and therefore reaching or not reaching a threshold. From the results in time period 3, deduce the nature of the synapses between neuron C and D. Explain your answer. So, you can see there is A being activated here. This is the hardest one, by the way. I don't necessarily expect you to get this first time. A being activated here, along with B, was enough for an action potential in D before. So A and B was enough to activate D. But somehow A and C is not enough to activate D. We know A is positive then, because A and B together did activate D. We know A is positive. If C was positive as well, then we would have an action potential in D. C cannot be excitatory, it cannot be releasing acetylcholine. Why must it be then that it's preventing an action potential? It must be because it is inhibitory. It must be a GABA neuron or something like it. It must be causing chloride ions to enter D. So, more IPSPs than EPSPs. I don't need you to know those terms. They're a bit fancy. You can call them that if you want. Excitatory postsynaptic potentials, inhibitory postsynaptic potentials. Don't need to use those words. Reduces membrane potential or makes it more negative or allows hyperpolarization or cancels the effect of action potential from A. Any two here. So I would expect you to say that it makes it more negative or it hyperpolarizes it and therefore cancels, is not necessarily the word I would use, inhibits perhaps the effect of action potential from A. So the idea that C is not just not helping, is that it is actually actively hyperpolarizing D. It's causing chloride ions to enter D. It is a GABA or something like it neuron. Now, this is a relatively complex topic. It's on the boundary between what I would need to talk to you about for a live lesson and what I'm happy to do with a video lesson. So that if you are struggling with anything, I would like you to let me know via Teams. Just contact me or your teacher, whoever's teaching you this. So have a go at these questions. I'm sure you have already. If you're struggling with them, contact me and I'm happy to help. Anyhow, assuming you've got that summation, we've then completed 2 of the 3 parts of the neuron we're going to talk about. We've completed the main body of the neuron and how action potentials move along it. We've completed the synapse, which is how it transmits to new neurons. The third part we need to finish, we're going to finish next lesson. We're going to talk about how the whole thing starts off, i.e. the beginning of the neuron. We're going to talk about the receptor, i.e. the pressure receptor and the light receptor. Right, have a very good day, Year 13. I'm looking forward to seeing you for our live lesson at some point in the near future.